Okay, what are the nucleic acids actually? Nucleic acids are acidic molecules which are found inside the cell nucleus, okay, which found in the cell nucleus. So the function of the nucleus is what, Jahangir?
The function of the nucleus is of the nucleus, yes?
Yes.
So the function of the nucleus is to control center of the cell.
This is the control center of the cell. What else, Munisa?
Has genetic materials, DNA,
Mm-hmm.
and it's bound by a double membrane.
And bound by double membrane. Okay, so the word nucleic tells you that they are found in the nucleus, right? There are two main types of the nucleic acids. The first version of the nucleic acids are DNAs, okay? They are DNA. And the second is RNA, okay? So DNA and RNA. So DNA means the full name of the DNA. You have to know deoxyribonucleic acid, okay? DNA is just abbreviation. So the full name of the DNA, deoxyribonucleic acid. The full name of the RNA is ribonucleic acid, okay? These are the molecules that store and transfer genetic information in the every living cell, okay? In the every living cell, DNA and RNA, they store molecules, okay? Or genetic information all the time, okay? So what they made of, what the nucleic acids are made of. So nucleic acids, so we know the monomers of the proteins, right? So that what's the monomer of proteins? So that
Acids.
proteins, the monomer of proteins. Jahangir?
It's different types of acids,
Which acids?
especially for protein.
It's a monomer of proteins, Munisa.
Amino acids.
Of course, amino acids, the monomers of the proteins, amino acids. What's the monomers of carbohydrates? Saudat? Jahangir?
So monomers of carbohydrates are it's a monosaccharides.
It's saccharides, just saccharides, okay?
Or
What's the
just
monomer of lipids, Munisa?
Like carbohydrates, or they don't have monomers.
The monomers of lipids.
Hydrocarbon.
Hydrocarbon, right? Okay, what's the monomer of nucleic acids?
Nucleotide.
Yes, nucleotides, okay? So the monomers, building blocks, monomers means the building blocks. The monomer building block of the nucleic acids are nucleotides, okay? They are nucleotides. If you link many nucleotides together, if you link a lot of nucleotides together, you get the polynucleotide, okay? As you can see here, this is one nucleotide here, okay? This is one nucleotide. This is another nucleotide. This is another nucleotide, okay? Nucleotide, nucleotide, nucleotide. So if you link many nucleotides together, you will get the polynucleotide. We call them deoxyribonucleic acid or ribonucleic acids. Okay, we say them like this. Okay, so the nucleotide also composed of three things. The first is sugar, the second phosphate group, and nitrogenous bases we have. Okay, as you can see here, the nucleotide, imagine this is one nucleotide, and this is one nucleotide. So in this nucleotide we have the sugar, we have the nitrogenous base. Okay, we have nitrogenous base, we have sugar, and we have the phosphate group. Okay, so we have the phosphate group, we have the sugar, and we have the nitrogenous bases. Okay, so the sugar can be in two types. If this sugar is located in the DNA, if we are talking about the DNA molecule, the name of that sugar, will be what in this DNA molecule? Deoxyribose sugar. Okay, deoxyribose deoxyribose sugar. Okay, deoxyribose sugar. If this sugar in the RNA molecule, we call it ribose sugar. Okay, it can be deoxyribose or ribose sugar. Okay, so deoxyribose in the DNA and the ribose sugar in the RNA molecule, and they have the phosphate group. Which three essential things in our body are composed of the phosphate? One of them is DNA or RNA. What else does have the phosphate? Phosphorus. Do you remember?
Like in ATP, energy
In the
storage
ATP and
transfer.
in the phospholipids. In the phospholipids as well, we have the phosphate group, right? In the DNA, in the RNA, we also have the phosphate group. And in the ATP, we also have the phosphate group. These are the essential molecules that contain the phosphorus. Okay, so sugar molecule, we have the deoxyribose sugar. We in the DNA, ribose sugar in the RNA, and additional thing we have here the phosphorus. Okay, and what else we have? We have the nitrogenous bases. What are the nitrogenous bases? Nitrogenous bases can be in four types in the DNA. They can be as a guanine, they can be as a cytosine, they can be as adenine, they can be as thymine. Okay, so nitrogenous bases can be guanine, cytosine, adenine, and thymine. Okay, for the
Yes.
nitrogenous bases. But for the RNA, if it's in the RNA, we will have guanine, cytosine, adenine, and uracil. Okay, not thymine. They have uracil. Okay, they have the uracil instead of the thymine in the RNA molecule. Okay, any questions? And you shouldn't forget that adenine, always adenine is going to link with thymine. Okay, guanine is always going to link with cytosine. Okay, okay, and sorry, it should be guanine, it should be cytosine, it should be adenine, this should be thymine. Okay, according to the formula. Okay, what's the difference? What kind of difference can you see between the cytosine and guanine? What kind of difference can you see?
Color difference.
Huh?
Color difference.
Okay, what else? Okay, here you can see only one ring in cytosine. Only one ring in the cytosine molecule. But in the guanine molecule, you can see how many rings we have? We
Two
have
rings.
two rings. Okay, which one is bigger, cytosine or guanine?
Guanine.
Guanine. Let's look at the thymine and adenine. Here as well, in the thymine we only have one ring, but in the adenine we have the two rings, right? That's why we divide the nucleotides according to the rings into two types: purines, purines, and pyrimidines. Pyrimidines. So example for the purines are adenine and guanine. They have the two rings, and they called purines in biology. And we have the pyrimidines. Example for them, one is cytosine, the second is thymine. Okay, so guanine and adenine, they have two rings. They are purines, and cytosine, thymine, they have only one ring. They are pyrimidines. Guanine always going to bind with cytosine, thymine always going to bind with adenine. Okay, and between them, I have to mention, between the thymine and adenine, they have how many hydrogen bonds? As you can see here,
Only two.
yes, between the thymine and adenine, we have two hydrogen bonds. Between cytosine and guanine, we have three hydrogen bonds. Three hydrogen bonds. Okay, good. And always remember, the number of the cytosine always equal to the number of the guanine in the DNA molecule, not in the RNA, okay? In the DNA molecule, the number of the cytosine always equal to the number of the guanine. And the number of the thymine always equal to the number of the adenines, okay? Because they are complementary to each other. If you have 100 thymines, you will have 100 adenines because they're complementary, because they come together. If we have 100 cytosines, we will have 100 guanines in another. If in one part of the in one strand of the DNA, if we count and calculate, if we have 10% of the cytosine in one strand of the DNA, in the second strand of the DNA, we will also have 10% of the guanine, and others will be thymine, others will be adenine. Okay? Good. Any questions?
No questions.
Okay, what's the difference between the DNA with RNA? So we have the DNA molecule and we have the RNA molecule, right? What's the difference between them? DNA is how many strands of DNA? We have two strands. This is the first strand, this is second strand. We have two strands of DNA. What about in the RNA? We only have one
Only
strand. Okay, only one strand in the RNA, two strands in the DNA molecule. Okay, one strand in the RNA, two strands in the DNA. What's another difference? Another difference in the DNA, we have adenine, thymine, guanine, and cytosine, right? But in the RNA, we will have adenine, guanine, cytosine, and instead of thymine, we will have the uracil. Okay, what's another? In the DNA, we have the the deoxyribose sugar. In the RNA, we have ribose sugar. Okay, so this is the first difference. First difference in the strands. Thymine is second difference with uracil. Okay, what about the third? This is deoxyribose sugar, and this is ribose sugar. This is the third difference. Okay, good. Any questions?
no questions.
Okay, so sugar, they differ in the sugar, they differ in the base pairs, uracil in RNA, thymine in the DNA, and in the shape, in DNA we have 2 strands, RNA we have 1 strand, okay? And in the function as well,
Okay.
stores the genetic code. DNA stores the genetic code, but the RNA helps to make proteins, okay? So messenger, transfer, ribosomal RNA types we have, okay? So why it's important? You want to ask something, Jahangir?
No, no questions.
Okay, good. So what are the 3 parts of the nucleotide, Jahangir? Let's recap. 3 parts of the nucleotide. What are the 3 parts of them?
3 parts of the nucleotide.
Yes.
The
Phosphate
sugar,
group. Sugar.
phosphate group,
Mm-hmm.
then sugar and base.
And base, nitrogen bases, okay? Which sugar found in RNA, Munisa? Munisa, which sugar is found in RNA?
Ribose.
Okay, which sugar found in DNA? Saw that?
Deoxyribose.
Okay, Jahangir, which nitrogen base is only found in the RNA, not in the DNA?
Only in RNA. Uracil.
In the DNA. Sorry, in the
In the
RNA, right?
DNA.
I asked it. If it's in the RNA, yes, you're right, the uracil. If in the DNA,
Thymine.
yes, thymine. Okay, good. So, Munisa, why is DNA called double-stranded while RNA is called single-stranded? Why? Munisa?
Because DNA has 2 hydrogen bonds.
So, because DNA has 2 strands, right?
Yes.
They
I
run
remember.
they run opposite to each other, okay? One strand goes in this way, another strand will go to the opposite side, okay? So it has 2 strands which run opposite directions. We call them anti-parallel, okay, and twist against each other. But whereas the RNA is only 1 strand, okay, only 1 strand. That's why we call it double-stranded, and RNA is single-stranded, okay? So my question to Saudat, what determines the genetic code in the DNA molecule? What determines the codes in DNA molecule? Adenine, thymine, guanine, and cytidine. They determine the code, okay? Determine genetic code. We will learn the genetic code later, okay? So this is your 5th week, right? This is your 4th week. This is your 4th week.
4, yes.
4th week, and you will have the 5th week, and after that you will have the midterm exams. You should know for it, okay? What's the function of the RNA in the cell, Jahangir?
Function of RNA in the cell.
Don't forget, okay? The function of the
Thing.
RNA to help to make the proteins. They make the proteins by making messenger RNAs, by making the tRNAs, by making the ribosomal RNAs, okay? We have 3 types of the RNAs: messenger, transfer, and ribosomal RNA, okay? Okay, good. This is a nucleotide diversity. Here it's illustrated. Here you can see in this image, right? What can you see in this image?
Pyramids.
See?
Yes.
Purine,
Pyramids.
pyrimidines,
Not
bases,
proteins.
nitrogenous bases, right? All of them. So we have the four possible DNA bases, right? So adenine, guanine, cytosine, thymine, right? This is four nitrogenous bases. We call them adenine, A, G, C, T, adenine, guanine, cytosine, thymine. Okay, these letters are genetic codes. Genetic codes. Okay, the genetic code, just like letters, make words in sentence. Okay, so these are the letters and the full uh, name of those letters is written next to them. Okay, so DNA nucleotide structure, we learned about it, right? Each DNA nucleotide it has three parts. As you can see here, it has three parts. First is sugar. Sugar, it can be in two parts. The first is deoxyribose, it can be, or it can be the ribose, right? It can be in two types. In the ribose, you have what? You have extra oxygen. Compare it to the deoxyribose. In the deoxyribose, you removed one oxygen. The difference between the ribose and deoxyribose, lack of one oxygen. Okay, so what else? In the nucleotide, we have the phosphate group as well. Okay, this is the phosphate group, and we have the nitrogenous bases, right? Nitrogenous bases, we have adenine, guanine, cytosine, thymine, uracil. Okay, cytosine, thymine, or uracil, they are pyrimidines. Purines are adenine and guanine. Okay, what's the difference between the adenines and guanines? Who can answer?
Like in guanine, we have oxygen
What's
instead of.
the structural difference? In what structural way
Yeah.
they differ from each other? Purines have two rings. In the purines, we have two rings. Adenine and guanine, they have two rings. In the pyrimidines, we have only
Only one.
one ring. Okay? Which DNA molecule is bigger? Those who contain more purines or those who contain more pyrimidines? Which DNA molecule will be bigger? Those who contain more pyrimidines or those who contain more purines?
Purines.
More purines means more size because they have extra ring, right?
Yeah.
Okay. How can you remember the purines as an adenine and guanine? You can learn a mnemonic like pure as gold. That means purines are A as gold. A, this means adenine, and G means guanine. Okay, so pure is as gold. Pure as gold. A, adenine, guanine, P means purine. Okay, pure means purines. Okay,
Yeah.
good. So you can remember adenine, guanine as purines. So, any questions? Jahangir, what are the four nitrogenous bases found in the DNA?
Four nitrogen bases found in the
Mm.
adenine, thymine, guanine, and cytosine.
And
Cytosine,
cytosine. Okay. So, Munisa, which base is replaced in RNA? What's replaced with?
Uracil.
Replaced with what?
Uracil.
Yes, uracil replace it with what?
Thymine.
With thymine. Okay. Okay. Any questions?
No.
This is the structure of the DNA, and this is the information regarding the who discovered the DNA. Okay. So the person who discovered the DNA is young. He was born almost 75 years ago. It was discovered by Watson, Crick, and Franklin. That's why they were rewarded with Nobel's Prize. So DNA, as you know, is made of two strands twisted together like spiral, staircase, or ladder. It looks like a ladder. This shape is called double helix. You know it. It was discovered in the year 1953 by Watson Crick and importantly Rosalind Franklin, okay, who contributed key X-ray images. Franklin also contributed to the key X-ray images. He discovered the X-ray as well. So DNA as a ladder, this is the analogy. So made of alternating sugar and phosphate groups, okay, this part stays the same in every DNA molecule. In each DNA molecule, the sugar and the phosphate, this is the part of the nucleotide, they are the stable, they stay the same, okay, these backbones of the DNA, they stay the same, those backbones. So sugar phosphate backbone, they are the same here, same in here, okay. So that's why they're called the backbone. This is just the backbone. This is just the uh, uh, what the strand is made of. The strand of the DNA or RNA is made of, okay? So, but what can be the variable? What can be changed? Here, as you can see, nitrogenous bases can be changed, right? Nitrogenous bases can be changed. It can be either adenine, guanine, thymine, adenine, cytosine, we don't know, okay? Between cytosine and guanine, we have 3 hydrogen bonds. Between adenine and thymine, we have 2 hydrogen bonds, okay? So, um, that's it. Okay, so you should know about it. The backbone is made of phosphate and the sugar, and base pairs are variables. Okay, what shape of the DNA? The shape of the DNA is double-stranded DNA's shape is like a ladder, discovered by Watson and Crick and Rosalind Franklin. Okay, Jahangir, what forms the sides of the DNA ladder? Sides, 2 sides of the DNA. What it is formed by?
I think it's formed by the by base
By sugar
or no?
and
Oh, but yeah, by sugar
and
and carbon.
phosphate. By sugar and phosphate. You see, this is the sides of the DNA ladder. These are 2 sides. In the sides, you can see only sugar and phosphate molecule. Okay, sugar, phosphate. But in the center, you only can see the nitrogenous bases. Okay, the base pairs. Good.
Yeah.
Any questions regarding this? And DNA base pairing. Jahangir, what do you know about the DNA base pairing?
DNA base pairing. Maybe by the picture I can see that it's something that matches.
Adenine always matches with what?
Thymine,
Thymine. Guanine always base pairs with
cytosine.
cytosine. Okay, good. If one strand is in the DNA, if one strand is adenine, thymine, and cytosine, then another strand will be thymine, adenine, and guanine. Okay, they complementary. Adenine always complementary to thymine, thymine complementary to adenine, cytosine complementary to
guanine.
guanine. Okay, between adenine and thymine we have 2 hydrogen bonds. Between thymine and adenine also 2. Between cytosine and guanine we have 3 hydrogen bonds. Okay, you shouldn't forget about it. Okay, good. Any questions? Any questions so far?
I get no questions.
Okay. So here it's illustrated how the DNA is anti-parallel to each other. What's the meaning of anti-parallel? If one strand is going to the left side or right side, the another will go to another place. Okay, so they are anti-parallel to each other. They are not like this. Okay, they're not like this. They are anti-parallel. Okay. So in the DNA double helix, the 2 strands run in opposite directions. One strand run from the 5' to 3'. Another is going to go to the from 3' to 5'. What does it mean, these numbers? What's the meaning of 5'? What's the meaning of the 3'? Okay, so what's 5'? This is the this refers to the carbon position in the sugar molecule. Okay, this is the sugar. Which sugar is that? Saudat? Saudat, what's the name of this sugar?
Guanine.
Koyman sugar diyorum. I'm saying sugar. Jahangir?
Um, you're asking to the
What's the name of this sugar?
Deoxyribose.
Deoxyribose. Why it's deoxyribose? Because it's DNA molecule. Because this is DNA. Here we have 2 strands, okay? The backbone is made of what, Jahangir? What is this backbone is made of?
of a sugar.
And phosphate. Okay, only sugar and phosphate. Why we are saying here 5'? Because this phosphate is attached to the 5th carbon of sugar. Here, the 5th carbon of the sugar. Okay, understood?
Yep.
This is the 5th carbon. This is the 4th, 3rd, 2nd, and the 1st. Okay, so this is the 5th one. What about here? What about here? Here attached to the 3rd carbon. Okay, attached to the 3rd carbon. Will be sorry. 2nd one, 3rd, 5. Okay. So here this phosphorus is attached to the 3rd carbon, so we call it 3'. So it can go either from the 5' to the 3' or from the 5' to the 3'. We don't know, from 3 to 5. And where the 5' located, to another strand will be 5'. 3' in one strand, another strand will be 5'. Okay, good. Any questions? So DNA bases, for example, the adenine, thymine, cytosine, guanine, must pair evenly and securely through the hydrogen bonds. Okay, so here you can see the hydrogen bonds between adenine and thymine, hydrogen bonds between cytosine and guanine. For this to happen, one strand must line up in opposite directions like a zipper teeth. Okay, for this happen, the strands must line in opposite directions. It's like a zipper. So this is how the DNA achieves the stability and proper copying. Okay. So 3 prime and 5 prime, the lane signs on the road. Okay. Like a lane signs on the road show direction of the strand. Okay, 5 prime and 3 prime shows the direction. Anti-parallel, that means two, for example, two escalators next to each other. One escalator is going up, one escalator is going down. The DNA is in this analogy. Okay, so why we need it? So it keeps the DNA base pairing more aligned. Okay, so sugar backbone, what is it? It's the side of the two letter. This is the backbone, sugar phosphate backbone, because in the backbone we have the sugar and the phosphate molecule. So, and we have the steps of the ladder. These are the base pairs, adenine, thymine, oxytocin, guanine. Okay, good. Any questions regarding this? Uh, here as well, you can see the three hydrogen bonds here and two hydrogen bonds here, right? So any questions, guys, regarding the nucleic acids? So Jahangir, read the first one. You don't have questions?
The fact that double standard DNA forms a double helix was discovered by, I don't remember the second name.
Watson, Crick, and Franklin. Okay, so Watson, Crick, and Franklin. Okay, second one, Melissa.
The four DNA nucleotide bases are adenine, thymine, cytosine, and guanine.
Third one, saw that.
RNA is not a double standard molecule.
Fourth one, Jahangir.
RNA nucleotides do not the same basis as DNA nucleotides.
What's the difference in the RNA?
Uracil, uracil.
Yes, uracil. Okay, fifth one, Melissa.
In DNA, guanine forms a base pair with cytosine, whereas adenine forms a base pair with thymine.
Okay, sixth one, saw that.
The nucleic acid backbone is made up of sugar and phosphorus, or I don't remember.
Yes, phosphate group. Okay, seventh one, Jahangir.
Sugar DNA is a deoxyribose.
Eighth one, Melissa.
In RNA, adenine can form a base pair with uracil.
Yes. Okay, good. Good. I hope you understood everything. Thank you. So we finished the macromolecules. Congratulations. Okay. Any question so far from biomolecules?
I get no questions.
No questions.